So one of my closest friends is actually a journalist, and he always told me when you have to tell a story or an event, just make sure to cover the 6 Ws, so the what, how, when, who, where, and when. I remember them all. And so I will use this frame basically to tell you the story of Zalmoxis from inception all the way into the approval and clinical commercialization. And hopefully within each of this area, I will show you what were the challenges that we had to face and the lesson that we learned on that journey. So let's start with what. What is Zalmoxis? Basically, it's a classical gene therapy approach, Ex-Vivo, that was developed starting in the early 90s, so 25 to 30 years ago, when they were coming together on one side antiviral therapies like acyclovir, ganciclovir, that were able to recognize a specific viral protein and selectively kill the infected cells. On the other side, you all remember that in those years, all this DNA technology were invented and were developed, so allowing for the gene modification, vectorology, and so on and so forth. So the idea behind Zalmoxis was, how about we take this very selective viral protein and we make it express on, instead of in infected cells, on a cell population that can potentially lead to lethal side effect like graft-versus-host disease, and then we activate that as a suicide gene in a case of GVHD itself. So basically, that was implemented in a setting of haploidentical transplantation. So those mismatch transplantation, where if you transplant the T cells from the donor into the patient, there is a high risk of lethal effect, and that's where you need the suicide gene. So that's where the Zalmoxis were invented. Second in line is the how. So how do you manufacture this kind of therapy? And that's basically where all the challenges began. An ex vivo gene therapy, as you all know, is basically made up by two big pieces, viral vector and cell manipulation. So viral vector, when you attend this kind of conference, you hear just one word all over again, and it's scale, scale, scale. And back 25 years ago, early 90s, we already knew that that was the direction. Great vision. We had implemented already there a bioreactor, a 100-liter bioreactor, a big piece of metal. And that was suitable for actually producing vector for phase 1/2. But it had, at the time, two big limits that are, on one side, the fact that they didn't exist, the scale-down model. So at each time you do some development work, you need to run the full scale with limit in economics, with limit in timelines. So that wasn't really suitable for doing any real optimization. But second, and the most important limit of that piece of equipment was that it was not disposable. And that was just a nightmare for doing the cleaning, sanitization, and for manufacturing multiple vector out of the same equipment. So although we did use that for phase 1/2, it wasn't suitable to continue that long. So we went back to what at the moment was the standard, that were disposable plastics. So roller bottle first and cell factories right after. And that's still the platform that is in GMP at the moment. But finally, a few years ago, technology evolved, and now we finally have disposable bioreactor, like the one that we are using, the Pol-iCellis, for example. This does have a scaled-down model, and this is disposable. So we can now use them for the purpose that we had in mind 20 years ago, but when the technology wasn't really ready yet. Second piece of the process is the cell manipulation. Again, when you do, and you go at this kind of conference, you just keep listening all over, closed process, automation, close the process, and automation, right? So again, we had a vision 20 years ago of closing the system, okay? And I even came across a 2001 business plan very recently that my predecessor made. And in that business plan, there was a very futuristic business model where MolMed was kind of supplied the clinical center with viral vector, and the clinical center was equipped with an automated machine able to do a decentralized manufacturing. So this is something that now many companies like Miltony and GE and Lonza are postulating, and we had that in mind way before it became now as a real actual model. So going back to this vision, we, at the time, none of these instruments were available. So we engaged an Australian contractor to build our own all customized solution. And we had a long discussion with them. We even made a model and, well, actually a real prototype. But our science, we were the scientists in this collaboration, and they were the engineers. The limit is that we couldn't really talk, and there were so much gaps between the two competences that it just didn't work through. So the successful story now out of the new provider is that they really provide and are able to connect science and engineering, something that we at the time weren't able. So again, we had a vision, we tried it to implement it, we didn't succeed, so we went back to the standard open process that is still the approved process at the moment. But finally, again, a few years ago, Miltony, Prodigy, and other instruments came back, and so that's where we restarted picking up our vision, and finally we're able to implement that. And so we are now working to finally close and automate the process. So the take-home on the first point, that how, is really, it doesn't really, you can have the best idea and the best vision in the world, but if you don't have the ecosystem around you, it's not going to be delivered, and it's not going to go substantially anywhere just because you alone are not really able to build everything you can possibly need to bring this kind of therapies to the market. So moving along, next will be why. Why would you, why did us develop Zalmoxis? And the answer is always easy: patient and medical need. Unfortunately, medical need and standard of care doesn't stay still. It moves, it evolves. And sometimes it evolves not out of the company, but out of the clinical practice. So something that you don't see as a competition then becomes your real competition. So when we started Zalmoxis development, the standard of care was very easy. So when you have a mismatched donor, you can only transplant stem cell. Because if you also transplant the immune system, that immune system has such a high risk of inducing graft-versus-host disease that you just prefer not to use the immune system and live with a high risk of leukemia relapse and infection. So using Zalmoxis at that time and being able to transplant also the immune system had a lot of benefit on that kind of protocol. But nevertheless, when you have a huge medical need, there are people actually working on solving that issue. And that means that clinicians in the last 20 years initially developed a new protocol called cord blood or double cord blood that I'm not going to enter into details, but basically try to address some of those limitations. After a few years, unfortunately, they realized that it didn't really solve the problem. So nowadays, it's the practice of cord blood is still limited. But again, a few years after, and we are now in the mid, well, I would say 2010, 2011, new protocol came in, the so-called T-repleted protocol, where you use the immune system, but you give cyclophosphamide and other immunosuppressant to control the acute graft-versus-host disease. In that case, of course, there are, again, limitations to that protocol, but the main side effect, that is the acute GVHD, is control. And so clinicians are liking it, and that's becoming the new standard of care, and this is here to stay. So what was the clinical need when we started wasn't there to the same extent when we finished. So we had to evolve in terms of adapting our clinical trial, adding a control arm, and also adapting the design of our studies in order to fit this evolving clinical need. And very importantly, and I think the challenge here is that if you are developing such a transformative therapy with a high cost of good and for which you are definitely seeking a high price, you need to make sure that you are delivering a transformative clinical benefit. Otherwise, the whole system is not just sustainable. And in that case, you need to see where other approaches are coming and monitor them. Otherwise, you will end up with a product that is not really commercializable just because it doesn't solve a new medical need. So next in line will be the where. Where do we manufacture this product? And again, try to remember what was like in the 90s. So there was no CDMO around, no facility, at least commercial facility, maybe some academic GMP facility here and there, but nothing as close as what we have today. That is still, by the way, under capacity to the current need. So there we had only one solution. It was just build our own facilities, and that's indeed what we did. We were the first one in Italy, but I think also in Europe, to get the first ATMP certification, and that was back in 2003. And we built it, and not only one facility, but once we hit phase 1-2 with positive result, we realized that that wasn't enough to potentially support commercialization. And that's where we start building a second facility. Then for the challenges that I partially already explained, and I'm going to explain in the next few minutes, we ended up having a slower ramp up than expected. And so we were in a position where we had excess capacity. We had a lot of GMP knowledge. So the easy solution will then to use that as a CDMO. And indeed, that's where, at the moment, we are even more famous as a CDMO rather than a developer, really because we could leverage on all this expertise and capacity and to put in the service of somebody else developing these therapies. Next point will be the who. If you want to develop a product like that, you need to make sure that you have resources available. And especially for GMP manufacturing and for development, you need to source qualified people. We were lucky enough to be located in Italy. That is not a good luck for many aspects, but for people, it was. We were in an environment where all this ex-vivo gene therapy were actually being developed with people like Bordignon, our founder, Maria Grazia Roncarolo, that yesterday got awarded, Igi Naldini, Fulvio Maviglio, and many others. So by chance and by them building up on their expertise and entire environment, we ended up with several universities and institutes nearby Milan for which we could access resources for building a very qualified team able to bring these therapies along this journey. Then there will be the when. When it's another very important aspect. When it's evolving, it's when we started in the late 90s, gene therapy was at the beginning. We were pioneer, very few companies working in this space. And everything went pretty nicely until the late 90s when probably you all remember the trial of Cavazzana-Calvo and the insertional mutagenesis induced the leukemia. And that kind of put the entire cell and gene therapy field a little bit on hold. And that in that time it was really hard to finance if you were a pure cell and gene therapy players. And that's where we needed to differentiate a little bit our portfolio in order to continue the development. So we became more an oncology company rather than a pure cell and gene therapy. We added another asset, a recombinant protein. We brought that throughout phase 3. Unfortunately, in phase 3, we didn't have the expected result. So that product didn't achieve the commercialization. But in the meantime, the industry changed. GSK entered into the field, Novartis entered into the field, and so cell and gene therapy kicked back in. And that's where we could go back to become once again, a pure player in cell and gene therapy. And now, once again, we are in a position where financing is becoming easier just for cell and gene therapy because of the environment. So once again, the point is, it doesn't really matter what you do, but you have to look at the overall picture to be successful. And timing is particularly crucial, and here is a little of an anecdote. For example, when we got the approval, CHMP approval, that was we were all partying on the night, and we woke up the next day hoping to have 50%, 100% increase in our share price, as you will expect once you get the approval. At the same day, UK voted out of the union, so we ended up actually losing the share price in that occasion. And now it's even more funny because UK seems to want to get back into the union, so it's just affected us. Yeah. So basically, once again, I just wanted to summarize the whole story. It's been quite a journey. We face a lot of challenges on the process side, on the when and the environment, on the where, the facilities, and you heard all what we went through. But at the end of the day, we are still a company with a great vision that weren't supported by the environment. We were sufficiently flexible to realize and to use the asset that in the meantime we developed, like the facility, the expertise, to use that as a CDMO. And so now, and not only for the CDMO, but also to this lesson will be crucial for the remaining pipeline that, by the way, we have. We are still active in autologous CAR-T, and we very recently opened up an allogeneic approach using CAR-NK. So all these lessons are now the basis for our new pipeline and for our CDMO business and for the future growth of the company, although Zalmoxis have been quite a journey with several challenges in this road. Thank you.